Hello, and welcome back to SKDB1's review on another student project that we have been working on recently regarding the topic of Dogma Central in Molecular Biology. As usual, before we start our presentation, I would like to introduce each of our group members individually. Unfortunately, I can only show them in slides because our time is very limited. But don't worry, because before each of our presentation, we will introduce ourselves once again. The first six of our group member will be Geg Mas, Danissa, Marvella, Karin, and Satria. The other six of our group member will be Julia, Krishna, Devi, Librawan, Jane, and Bintang. And last but not least, it is myself, Aryana, as the group leader of SGDB1. In this presentation, we will discuss this topic by dividing it into 5 subjects. As our topic say, the first subject will be mainly discussing about Dogma Central and its history. The second subject will be mainly about biomolecular review on our genetic component. And finally, the third, fourth, and fifth subject will be discussing about each part of the Dogma Central, which is replication, transcription, and translation. First of all though, it will be a much better choice if we talk about on our reasoning on why we choose this topic as our research. So my friend Devi will give you more insight about our background information, problem formulation, purpose, and benefits of this research.
Hello, my name is Devi Prastiti with number 21025110028, and I would like to explain a little bit about the background information for our student project regarding central dogma. So basically, every single organism on Earth has their own genetic code, which includes definite information. From a biological aspects, humans are the best at adapting to the change of environment by remaining predominant through some genetic process called protein synthesis. This protein synthesis includes the role of DNA. The double helix model of DNA, or deoxyribonucleic acid, was first found by James Watson and Francis Crick in 1953. The DNA can also be called genetic molecule, which is unique because of its nucleic acid content that has the ability to carry genetic clues from one generation to the next generation. And with that ability, DNA also has the capability to replicate itself, and in addition, those genetic clues can also be transcribed into RNA and also translated to the form of amino acids. They then experimented with the DNA and found out about genetic information with the correlation between DNA, RNA, and protein. The correlation displayed the conception of genetic code, which enabled Francis Crick to develop a thought of the link between gene and protein. The concept was named the flow of information. And later in his notes, the concept of this genetic flow is entitled Dogma Central. By what have I described, then raise a multiple question. First one, what are the definition and the history of Dogma Central? Second, which genetic components are involved in Dogma Central? Third, how does the process of DNA replication occur? 4, how does the process of transcription occur? And last, how does the process of translation occur? In this student project, there are several objectives that we should achieve. Generally is to learn more about each of the Dogma Central and their respective mechanism. Specifically is to describe the definition and the history of Dogma Central, to describe genetic components that involve in Dogma Central, to describe the process of DNA replication, describe the process of transcription, and also describe the process of translation. And by doing this student project, we as a group could learn the clinical sciences regarding the central dogma from various credible sources, and it could help the improvement of medicine, especially COVID-19 and its complication. And we also could learn more about the clinical knowledge required as a medical professional for undergraduate students. And the next chapter will be continued by my college. Thank you.
Hello everyone, my name is Putu Bintang Kirana with last name number R033. I will explain about the Dogma Central definition and its history. So Dogma Central is a fundamental principle about genetic information expression. It is referring to a molecular biology procedure in which genetic information is transferred from DNA to RNA, resulting the production of a protein molecule. As we can see in the slide, there are DNA replication, transcription, and translation. So those three have been defined as the core of the dogma system. Okay, next slide please. Thank you. So the concept and the model of the dogma central were created by Francis Crick and James Watson in 1953. Then in 1957, Francis Crick presented the dogma central schematic. He tells us about genetic information might be transmitted from DNA to nucleic acid and subsequently to protein. However, the genetic information cannot be sent from one protein to another or from one nucleic acid to another. Then in 1958, the notion of dogma central was introduced following the lecture. Okay, that's all from me. The next part will be explained by my friend. Thank you.
My name is Putu Krishna Arsawardani Putri with last digit number 027, and I'd like to explain about the DNA. DNA, or deoxyribonucleic acid, is a double helix structure with two polynucleotide chains. Every nucleotide monomer consists of a nitrogenous base, phosphate group, and a 5-carbon sugar called deoxyribose sugar. There are 4 types of nitrogen-containing base in DNA, namely adenine, cytosine, guanine, and thymine. The phosphate and deoxyribose sugar are linked by covalent bonds and form a backbone-like structure on the outer side of the double helix that provides DNA with structural integrity. DNA are antiparallel, which means the 5' and the 3' ends in each strand face the opposite direction. The 5' end of the DNA binds with phosphate group and the 3' end binds with hydroxyl group. The two polynucleotide chains are linked together by hydrogen bonds between the nitrogenous bases inside the double helix structure. The nitrogenous bases are paired by specific combination. Adenine is paired with thymine by forming 2 hydrogen bonds, and guanine is paired with cytosine by forming 3 hydrogen bonds. DNA carries biological information through the order of nucleotides along the DNA strand. This information must be copied for replication and transmission to the next generation when the cells divide into daughter cells.
Hello, doctor and friends. Today I'll be giving 2 materials in this student project. But before that, I'll introduce myself first. My name is Marvella and my last 3-digit number is 007. Now I'll be moving on to the explanation. RNA, or ribonucleic acid, is a polymeric molecule that is needed in the biological roles, especially in the dogma central. RNA is made from the nucleotides including a 5-carbon sugar backbone, a phosphate group, and nucleobases that are attached by the phosphodiester bonds, and it forms strands. Those nitrogen bases are adenine, guanine, cytosine, and uracil. There are 3 types of RNA, such as mRNA, tRNA, and rRNA. RNA's main function is to create proteins using translation and encoding. Maybe that's all from me, and I'll be back later for my next material. Thank you.
Thank you for the opportunity. My name is Putu Mas Isyundra, last digit number 003. Now, me and my friends will explain about the process of transcription. But firstly, I'll explain about molecular components. First is RNA polymerase. RNA polymerase is responsible for separating the 2 strands of DNA and joining RNA nucleotides as they form base pairs along the DNA template. Like DNA polymerase, which functions in DNA replication, RNA polymerase can assemble polynucleotides only in the 5' 3' direction. However, unlike DNA polymerase, RNA polymerase is able to make chains from scratch. These enzymes do not need a primer. Next are promoter and terminator. The specific sequence of nucleotides along the DNA can mark where the gene transcription begins and ends. A promoter is a DNA sequence to which RNA polymerase attaches and initiates transcriptions, while the terminator is a sequence that marks the end of transcription. Molecular biologists said there are 2 directions of transcriptions, namely downstream and upstream. This term is used to describe the position of nucleotide sequences in DNA or RNA. Thus, the promoter sequence in DNA is said to be upstream towards the terminator. The stretch of DNA that is transcribed into RNA molecules is called a transcription unit. That's all from me. Thank you.
Thank you for the time. My name is Nyoman Sujana with the last 3 digits of NIM number 011. So today, I'm going to explain to you about the mechanism of transcriptions. Transcription is the process of synthesis a new RNA. The process of transcriptions can be divided into three stages: initiations, elongations, and also terminations. Initiations. After RNA polymerase binds to the promoter, the polymerase unwinds the DNA strands and initiates RNA synthesis at a start point on the template strand. In eukaryotes, there is a complex called the transcription initiation complex, as you can see in the slide. Several transcription factors, when recognizing the TATA box, must bind to the DNA before RNA polymerase II can bind in the correct positions and orientation. Additional transcription factors, with purple color on the slide, bind to the DNA along with RNA polymerase II, forming the transcription initiation complex. RNA polymerase II then unwinds the DNA double helix, and RNA synthesis begins at the start point on the template strand. Elongations. The polymerase moves downstream, unwinding the DNA and elongating the RNA transcripts from 5 exons to 3 exons. After transcription has occurred, the DNA strands reform a double helix. A growing strand of RNA trails off from each polymerase, with the length of each new strand reflecting how far along this enzyme has traveled from the start point. In this stage, the base and the DNA are complementary to the new RNA. Purine bases turn into pyrimidines and vice versa. But there is something unique, namely adenine will be pairing with uracil in the RNA. Termination. Eventually, the RNA transcript is released and the polymerase detaches from the DNA. Termination mechanism started when RNA polymerase II transcribes a sequence on the DNA called the polyadenylation signal sequence, which specifies a polyadenylation signal AAUAAA in the pre-mRNA. Then, at a point about 10 until 35 nucleotides downstream from the polyadenylation signal, these proteins cut the RNA transcripts from the polymerase, releasing the pre-mRNA. The pre-mRNA then goes through processing before being a mature mRNA. As you can see in the slides, it consists of adding 5 exon cap modified of a guanine or G nucleotide added onto the 5 exons N. Adds 50 until 250 more adenine or A nucleotides, forming a poly A tail at 3 exons N. And last but not least, splicing and cutting out the intron in the pre-mRNA by spliceome. I think that's all from me. Thank you.
Thank you for the opportunity. First of all, let me introduce myself. My name is Adjani with the last 3 digits of student number 031, and I would like to explain about transcription factors. There are several transcription factors that are already known, which are referred to as general transcription factors or GTF. The first factors are transcription factors 2A or TF2A. It helps other Tata binding protein or TBP bind to the Tata box's promoter sequence. The interaction of TF2A with TBP will facilitate the formation and stabilization of the pre-initiation complex or PIC, while also prevents the binding of any repressive elements. The second one is TF2B, which acts as a link between TF2D and RNA polymerase II, while also be able to interact with BRE or B recognition element, a promoter element found in the Tata box. TF2B mutation causes a shift in the starting site of transcription. The third one is TF2D. It is the only GTF capable of recognizing specific DNA sequences or promoter sequence. TBP and various TAFs or TBP associated factors are its components. The fourth one is TF2E. It is thought to be involved in dissociating double-stranded DNA at the transcription initiation point. This protein has a zinc band motif that allows it bind to single-stranded DNA. And the last one is the F2H, which have functions to connect the nucleus to subcomplex kinases that activate cyclins, help to create replication bubbles, and implicated in the phosphorylation of amino acid serine in the terminal domain carbonyl enzyme RNA polymerase II, and perhaps other cell cycle proteins.
First, let me introduce myself. My name is Danissa Putri with 3 last digit number 005. Today, I will present about the mechanism of DNA replication. So DNA replication mechanism is first started at a specific place called the origins of replication. Protein that initiate DNA replication will automatically recognize this section and bind to them, causing the DNA strand to separate and open the replication bubble. A little fun fact, in eukaryotes, they have about 100 until 1000 of replication bubble, but in bacteria, they only have one replication bubble. Move on to the next mechanism. So at the end of the replication bubble, there is a section called replication fork, where the parent strand is being opened by the help of the helicase enzyme, as we can see from the picture. After the helicase enzyme opens the parent strands, there is a single strand binding protein that will bind into the single DNA and stabilize them, so the DNA will remain separated. Parent strand that is being opened is now available as template for synthesizing new complementary DNA strands. The result of starting nucleotides during the DNA synthesis is called primer RNA. New DNA will start at the end of the 3-axon RNA primer. An enzyme called DNA polymerase III is in charge of catalyzing the new DNA by adding nucleotides into the chain that already exists before. A little fun fact again, 2 DNA strands inside the double helix are antiparallel, which means DNA polymerase only can add nucleotides from the end of 5-axon until the end of 3-axon. We can see that too on the picture. Antiparallel characteristic resulting in 2 types of DNA polymerase adding mechanism. The first one is called leading strands, while the other one is called lagging strand. And now let's move on to the last mechanism. So at the end of DNA replication, there will be checking and repair phases that will be done by DNA polymerase. Then DNA polymerase will do some proofreading to check if there are any mistake. So that's all for me. Thank you.
Let me introduce myself. My name is Lanang Agung Librawan Laksana, with the last 3 digits of name is 029. I will explain the enzyme of DNA replication. DNA replication is a biochemical reaction. Enzymes are needed as a catalyst for biochemical reaction. Activation energy will be lowered by the enzyme, so the process is more likely to occur. There is a central enzyme involved, namely DNA polymerase, in the process of DNA replication. So, what are the other enzymes in DNA replication? The first one is DNA polymerase III. This enzyme works as the main enzyme in the DNA replication process, which is very important. The second is DNA polymerase I. This enzyme functions as a repair enzyme to eliminate error and fill in the gap in the order. The third is DNA ligase. This enzyme plays a role in sealing nicks in the sugar phosphate backbone. The fourth is RNA polymerase or RNA primase. The function of this enzyme is to make primary RNA. The presence of this enzyme will make DNA polymerase III can be sorted. The fifth is RNase H. The function of this enzyme is to remove the primary RNA when the role of the primary RNA has been completed. The sixth is DNA helicase and DNA gyarase. The function of this enzyme is to unwind the DNA duplex. The last enzyme involved in DNA replication is helix destabilizing protein or single-strand binding protein. The function of this enzyme is to prevent unwound DNA from rewinding immediately.
That's all I can explain about the enzymes of the DNA replication. Thank you.
My name is Julia Caroline and my last digit number is 016. The DNA synthesis that occurs along the two strands is different. The two strands are the leading strand and the lagging strand. It is due to the nature of the polymerase's synthetic activity, which is in the direction of 5 to 3 chain and anti-parallel of the two chains. The leading strand is made through the mechanism of DNA polymerase III, which synthesizes complementary strands continuously. The DNA synthesis produces a long continuous single strand. DNA polymerase III will remain at the replication fork and add nucleotides continuously to the complementary strand. A leading strand synthesized by DNA polymerase III require only one primer. The leading strand synthesized by DNA polymerase III cannot be continuous in one long section away from the replication fork, because DNA synthesis that adds new nucleotides only occurs at the growing end of the strand to the 3 end, the elongated DNA strand. Thus, the leading strand is synthesized in the form of small fragments called Okazaki fragments. The leading strand and lagging strand synthesis processes occur and are at the same speed. In 1953 to 1957, scientists debated how DNA could replicate. James Watson and Francis Crick in 1953 said that DNA consists of two helical strands wrapped around each other in a coil. Then, in 1957, three theories about DNA replication prevailed. The first theory is semi-conservative theory. According to the semi-conservative model, the old DNA strands are separated, and a new strand is synthesized on each of the old DNA strands. Then, two new DNA chains are produced, each containing the old molecule and a new chain. The second one is conservative theory. The old DNA strand will serve as a template for the new DNA strand, so the old DNA will be copied to form new DNA that is precisely the same without changing the old DNA, so that the old DNA would remain constant at each replication. The third is dispersive theory. Some parts of the original DNA chains will be used as templates for the synthesis of new DNA chains, so that at the end of replication, the DNA chains formed will contain the segments of old and new DNA chains. This replication produces two alternating old and new DNA molecules on each strand. After 2 years of research in the late 1950s, Matthew Meselson and Franklin Stahl designed an experiment on all three models. As a result, their experiment supported a semi-conservative model of DNA replication as predicted by Watson and Crick. That's all from me. Thank you.
Translation, as the name suggests, is a process of translating genetic information into protein. There are two unique molecular components required during translations, which is tRNA and ribosome. And I, Gede and Saputra, with last three-digit number of name 018, will discuss about one of them, which is tRNA. tRNA or transfer RNA is an AT base pair single-stranded RNase that is synthesized by RNA polymerase III in the nucleus that holds the important task of translating mRNA into polypeptide. This process is possible because of tRNA's very unique structure. As a single-stranded RNA, the presence of complementary base pair across its strand causes tRNA to form a loop-like structure called hairpin. And if this hairpin is flattened, it will take the shape of a lucky clover, as we can see in the slide. But in cytoplasm, tRNA assumes a three-dimensional L-shaped form that has two ends, one anticodon end and one peptide end. In the anticodon end, there is a specific sequence of base triplet called anticodon, which can be paired with another specific mRNA codon. On the other end, there is an overhang of the three-axon end of the RNA called peptide end, and this end can act as a binding site for the amino acids. Next slide, please. Okay, as we know, the process of translation is very specific. This accuracy is maintained by two methods unique to tRNA. The first method is regarding the specificity of amino acid that is being bound to tRNA's peptide end. This binding is facilitated by an enzyme called amino acyl tRNA synthetase that catalyzes the hydrolysis reaction between ATP-energized amino acid and tRNA. The said process produces amino acyl tRNA, or widely known as charged tRNA, and this tRNA is an tRNA that is bound to an amino acid via a high-energy ester bond. There are 20 different synthetases for each amino acid, where each type has a unique enzyme's active site for a specific combination of substrate, in this case tRNA and amino acid. Other than that, we know that the same amino acid can be carried by multiple kinds of tRNA, in which synthetase are able to detect all kinds of tRNA that coded the same amino acid, once again showing its incredible specificity. Next slide, please. The other method, the second method, is via the implementation of wobble codon. So here's the deal. If we count all the possibilities of nucleotide triplet combinations as anticodons, there are supposed to be 61 tRNA available. But instead, there are only about 45 of them, meaning some of them can share the same output with more than one nucleotide triplet combinations. This is later known as wobble codon phenomenon, where the third nucleotide in some anticodon is very flexible. They can bind into multiple base pairs other than the one they are supposed to bind. Example of this can be seen in the codon to peptide table, as we can see in the slide, in which the third nucleotide rarely influences the amino acid's outcome. As we can see, both AGA and AGG coded the amino acid arginine, AAA and AAG coded lysine, CAA and CAG coded glutamine, and many more.
Hi everyone, my name is Khadra Karin Kamila with the last 3-digit number 009. I would like to explain about translation in ribosomes. So, the ribosome is one part of cell organelles that doesn't have a membranes, and ribosomes have a role as a place for protein synthesis that can be found in prokaryotic and eukaryotic cells. Ribosomes translate mRNA to form a polypeptide chain using amino acids carried by tRNA in the translation process. Several steps in the manufacture of protein by the ribosome, namely, the first step is ribosomes attach to amino acids to build proteins, and the second, the large and small subunits of the ribosome are attached to the mRNA, and the third, ribosomes find a store region in RNA called codon. And the fourth, ribosomes go down RNA with each of the 3 letters in RNA representing a new amino acid, reading the instruction for the amino acid. And the last step is the stop code has been reached, then the protein formation process stops. The components of ribosome are rRNA or ribosomal RNA and ribosomal proteins called RNPS or ribonucleoproteins. The mass of the ribosome is about 65% composed of rRNA. Thus, RNA is the most abundant type of RNA. rRNA has the major role in the formation of subunits of the ribosomes. Ribosomes are divided into 2 based on their layout, namely bound ribosomes and free ribosomes. Bound ribosomes are ribosomes attached to the intracellular membrane, and free ribosomes are ribosomes that are present in the cytoplasm and float in the cytosol. The result of the free ribosomal translation will be transferred to the mitochondria used as ribosomal proteins or peroxisome enzymes. The translation results in a chloroplast, and mitochondrial ribosomes are polypeptides which will later be needed to meet the needs of both organelles. Ribosomes have a structure composed of 2 subunits, namely small subunits and large subunits, which when combined will form a subfunctional. Ribosome has one small subunit binding region called the A or amyocell, P or peptide region, as well as the E for the exit region located in the large subunits. Aminoacell tends to bind to tRNA that carries amino acid and will be added to polypeptide chain. Peptides tend to bind to tRNA attached to actively growing polypeptide chains. The E or exit site is the exit pathway for uncharged tRNA. That's all from me, thank you.
Thank you for the opportunity. My name is Geg Mas Anantalakshmi with the last 3 digits of name number 001. Today I will explain about the mechanism of translation. Translation is the process of protein synthesis in which the genetic information encoded in the mRNA is translated into a sequence of amino acids on a polypeptide chain. Translation occurs in 3 main stages. The first one is initiation. The first stage of translation involves the assembly of a ribosomal complex by 3 components. First, the small ribosomal subunit bind to an mRNA and an initiator tRNA, which adhere to the start codon. Then, the large ribosomal subunit join to complete the complex. The next step is elongation. Following the formation of the initiator complex, translation proceeds with the polypeptide chain elongation. Ribosome contain 3 tRNA binding sites denoted by the letter P for peptidyl, A amino acyl, and E exit. The P site is occupied by the initiator tRNA. The initial stage in elongation is for the amino acyl tRNA to connect to the A site. Following that, a peptide bond can be formed between the initiator tRNA and an amino acyl tRNA. The ribosome then moves 3 nucleotides along the mRNA. This phase moves peptidyl tRNA from the A site to the P site and an uncharged tRNA from the P site to the E site. The process of elongation and translocation are repeated as the ribosome moves along the mRNA sequence. The last step is termination. Polypeptide chain elongation proceeds until a stop codon is translocated to ribosome A site. This will induce hydrolysis of the polypeptide chain bond, leading to the release of polypeptide chain and the disassembly of the ribosomal unit complex. The next topic will be explained by my colleague.
Thank you for the opportunity. Back again with me, I'm Marvella, and my last 3-digit number is 007. After this, I'll conclude all the materials that have been given by my friends, and I'll give a suggestion regarding our student project. So the conclusion is, first, central dogma is a fundamental principle that explains the genetic information expression that results the production of the protein molecule. Next, the genetic components that are involved in dogma central are DNA, RNA, proteins, and transcription vectors. Third, the process of DNA replication occurs in majorly transcription and translation. Then, transcription is the process that is mainly about how the DNA is copied into mRNA. Occurs in 3 steps: initiation, elongation, and termination. Last, translation occurs when the protein or polypeptide is synthesized from the information that is stored by mRNA, and it also appears in 3 steps, such as initiation, elongation, and termination. It is highly suggested that all medical students should study about the dogma central. This way, medical students can easily understand about the role of the genetic information's flow and its function. That's all from me, and thank you.